I would encourage the groups that go forward. So I will give everybody the same feedback that I gave Ian last night, which was if you pick a very general article, spend some time in the primary literature that backs up the article. Like that WebMD article was written by a non-trained person. I saw it was reviewed by a doctor, but not written by a doctor. So if you do that sort of lay literature, make sure you delve into some of the papers that go behind it, as these guys did. So that was good. So hopefully there's going to be a little bit of review, but then we're going to get into a little bit more of the genetics of MHC. And it's a little bit overwhelming in terms of the amount of genetic material, but I'm just going to try to highlight some important themes and distinctions. So it's not a huge deal. So as we finished last class, cells that express class 1 are pretty much all the nucleated cells. Class 2 is professional antigen-presenting cells. Class 1 is intrinsic endogenous proteins. Class 2 is exogenous. Class 1 is to CD8, class 2 is to CD4, or cytotoxic T helpers. And then the class 1 has a transmembrane alpha chain and a secreted beta-2 microglobulin. And there are 2 transmembrane proteins for class 2. So in humans, the genes that encode class 1 and class 2 are in the HLA. It's called human leukocyte antigen gene complex. Regions A, B, and C each have 1 alpha chain gene per region for class 1. And then class 2 has DP, DQ, DR regions with multiple alpha and multiple beta chain genes per region. And then there are some other regions that contain class 1 and class 2 genes that are similar to class 1 and class 2 but don't present antigen. So they're pseudogenes or accessory proteins. So it's a huge gene family that encodes this functionality. So just looking at it on the chromosome, it looks like this. So it's a whole bunch of genes. Unlike the BCR and the TCR, there's no genetic recombination. It's just a bunch of similar copies.
I know they use HLA like proteins to match people for like transplants and stuff.
Yes.
Are there like specific ones they look to, or is it like all of them, or?
So that is the science of a person who does transplants, and there are major loci and minor loci. So it's very important to get the major loci matched and as many of the minor loci as possible. We'll have a whole class on transplantation, but because there are so many genes and not all of us have all the genes that exist in the population, it's extremely hard to match people for transplantation. But that's basically the gene family are named because they figured out the genes that are, before they understood antigen presentation, they understood how people needed to be related to transplant tissues and eventually isolated it to this region of the chromosome that determined transplant compatibility. Okay. So MHC is similar in the mouse. The difference with mice is that inbred mouse strains have identical MHC, which means you can transfer blood or tissues between mice of the same strain. There are certain MHC backgrounds that perform better in specific immune and infectious disease models. That makes sense because all of the peptide antigen presentation occurs through MHC. And the mouse MHC chromosomal region is synthetic to human, which means it's organized the same way, meaning that evolutionarily, some predecessor of mice and humans already had MHC encoded in its chromosome. Okay, so I wouldn't get too bogged down in the genetics, just there's a whole bunch of copies in the human population. We each have some combination of those copies, and we each express a bunch of different class 1 and class 2. So unlike T cells and B cells that have one TCR copy, one BCR copy, our cells express multiple MHC genes on the surface. Okay, so in terms of the protein, there's an N-terminal peptide binding domain that we've been talking about, and we'll look at the structure in a minute. Then there's the immunoglobulin domains that define the structure, and a single pass transmembrane domain. There's a short intracellular domain, so there's no signaling on the antigen presenting cell, only on the T cell side of things. And the affinity for peptide antigens is less than that of antibodies for their respective peptide epitope. So, and that's because, as we talked about before, each MHC protein needs to be able to fit a bunch of different peptides into it. So it's not a super high affinity interaction. So again, these aren't linear chains of proteins. They have both two-dimensional and three-dimensional structure. They have interchain disulfide bonds to define the regions, just like antibodies. And not surprisingly, the peptide binding domain is the most distant from the cell membrane. Okay, so class 1, there are multiple class 1 proteins expressed on every cell, multiple peptides processed from endogenous proteins. So, as we've been discussing, those proteins include viral proteins when the cell's infected. And except in the case of infection, there's almost no immune response because there's no TCR specificity for self-peptides.
The folding and assembly of MHC class 1 molecules takes place in the lumen of the endoplasmic reticulum. The initial folding of the class 1 heavy, or alpha chain, is aided by the chaperone calnexin. The partially folded chain is transferred to a second chaperone, calreticulin, which aids the further folding of the chain and the association of beta-2 microglobulin. Other proteins, ERP57 and tapesin, associate with the nascent class 1 molecule, which binds to the TAP transporter via tapesin to form a peptide-loading complex. The peptides that bind to the MHC class 1 molecule are generated by a large protein complex, the proteasome, which is found in the cytoplasm. The proteasome degrades proteins within the cytosol of the cell to produce short peptides, which are then transported through the endoplasmic reticulum membrane by the TAP transporter. Some peptides do not bind to the MHC molecule at all. Others can bind, but are unstable. These are released from the MHC molecule, a process called peptide editing. Finally, a peptide binds to the MHC molecule with high affinity to make a stable complex. This causes the final step in the folding of the class 1 molecule to take place, and the dissociation of the peptide-loading complex. The peptide-loaded MHC class 1 molecule is now free to exit the endoplasmic reticulum and be transported, via the Golgi apparatus, to the cell surface, where it can be recognized by the antigen receptors of CD8 T cells.
Just like that.
How long are the peptide sequences?
So when they're done being processed, they're 8 to 10 amino acids. They may be a little longer when they first load, and then they can be trimmed down by that BRAF enzyme we talked about last class. Okay, so the alpha 1 and 2 regions define the peptide binding domain, 8 to 10 amino acids loaded in each one. Each individual MHC protein, which is defined by the combination of two chains, can bind several hundred alternative peptides. You can imagine there's only, how many amino acids are there? 20 amino acids an 8 to 10 sequence, there's only so many permutations, but each protein can bind several hundred of them. The peptide association is determined by anchor residues at the end, so there's more flexibility in between. So there's some flexibility there. And then the alpha-3 region of class 1 compromises the association with beta-2 microglobulin. So this is a constant chain, and it's required for transport. So there are knockout mice for beta-2, and they have no class 1. So in these mice, you can transplant tissues from different mouse strains or even human hematopoietic tissue without rejection because there's no class 1 expression.
This is like 1 plus K mice can go.
Knockout. So if they eliminate beta-2 microglobulin, there's no class 1 expression. Okay, so this is what that looks like. Here's alpha-2 and alpha-1, and the red spiral is the peptide, and it's the interactions with the amino acids at the end of the peptide that define its binding in this particular MHC molecule. Okay. So for class 2, so this is peptides from the exogenous origin, not synthesized by the presenting cell. It's important to point out, we've been talking about how this is how we deal with extracellular pathogens, but most of the peptides are actually self, because the antigen presenting cells are picking up debris or apoptotic cells. The peptide group is similar to class 1, except that it's 2 proteins that define the region, alpha-1 and beta-2, and the ends of the groove are open rather than closed. The class 1 groove is open, the class 2 groove is open, the class 1 groove is closed, which is why you can then trim down different length proteins in the class 2.
The 2 chains of the MHC class 2 molecule assemble in the endoplasmic reticulum, the ER, with a 3rd chain, the invariant chain, shown here in green. A portion of the invariant chain binds to the peptide binding groove of the MHC class 2 molecule and prevents peptides or unfolded proteins present in the ER from binding. The invariant chain also guides the transport of the class 2 molecule out of the ER and through the Golgi apparatus into a vesicle that eventually becomes part of the endocytic pathway by which pathogens and foreign proteins are taken into the cell. Progressive acidification of this endocytic vesicle activates proteases that cleave the invariant chain in 2 places, leaving a small peptide, the class 2 associated invariant chain peptide, or CLIP, bound to the class 2 molecule. Engulfed pathogens or their proteins are also degraded by acid-activated proteases into peptides, but these cannot immediately bind to the class 2 molecule because the CLIP peptide still occupies the peptide binding groove. The removal of the CLIP peptide is the function of a specialized MHC class 2-like molecule, HLA-DM, which is also present in these vesicles. It functions as a catalyst, coordinating both the release of the CLIP peptide from class 2 molecules and the binding of pathogen-derived peptides. The MHC class 2 peptide complex is then transported to the cell surface, where it can be recognized by the antigen receptors of CD4 T cells.
Okay, so we've gone through that a few times. I won't go through it again, but basically that's what's happening in every T helper cell reaction for the rest of the semester. We talk about its response to a peptide antigen from MHC class 2. Okay, so the class 2 peptide groove is open and longer, so it can have longer sequences of amino acids. The peptides associate all along the length, not at the ends, so there's less flexibility. And so there are fewer alternative peptides can be presented by the same MHC. It has similar forces to antigen-antibody interactions like charge, hydrophobicity, and hydrophilicity. So looks quite similar to class 1, except it's open at the ends and longer. Okay. So compared to lots of other genes in the genome, there's huge diversity in MHC. And I think that was really highlighted by Jenny's question in the first lecture, why can't we just have one copy? And it's driven by 3 phenomena. It's driven by evolutionary pressure from infectious diseases, but the variability is encoded by 3 phenomena. One is polygenicity, so we have many copies of similar genes within each of us. Polymorphism, so there are many alleles in the population that are available, so each of us is a random combination of the alleles available from our parents, but not everything in the population. And there's codominance, so maternal and paternal copies of HLA genes are expressed within the same individual. So we're not just expressing our maternal ones or our paternal ones as we do with some other genes. Okay, so this is just a depiction of those phenomena. So there's polygenicity, so we've recopied and mutated the same genes many times. We have regions with different copies of the same gene, so a bunch of different class 1 alpha 1 copies. And the same region may have multiple copies of the same gene, so we may have a whole bunch of identical copies. And the composition of some of those regions are different between people. The alpha and beta regions from a single region predominantly pair together, so it's not like the B cell receptor and the T cell receptor where you can randomly pair up light chains and heavy chains. We're pretty much putting an alpha and beta from one region together every time. It's not random association. Okay, so within the population, there's more than 600 available HLA-A alleles, 900B and 300C, many alleles for class 2 as well. So as Marissa identified this, it's very challenging to match anybody perfectly for transplantation unless they are genetically identical. So for many genes, as I mentioned, either a maternal or a paternal copy is expressed depending on dominance. But for HLA, there are active mechanisms that make sure we maintain expression of both copies. So we inherit a whole chromosome from each parent, and we get a haplotype, an HLA haplotype from each of them. So there are ancestral haplotypes that were defined by the founders of isolated communities. So certain isolated communities have different frequencies of HLA haplotypes than the population as a whole. So the combination of polygenicity, polymorphism, and codominance results in the expression of many different MHC proteins on each cell. So a major difference from BCR and TCR. Class 1 genes are universally and constitutively expressed. Class 2 genes are upregulated during activation of immune cells. So an example, which I'll give in a minute, is some of my own data from human whole blood, that when it's exposed to TLR ligand, upregulation of class 2 can be detected in cells in the immune system. So I do a lot of lab work with whole blood samples from autoimmune patients. So I take that blood, stimulate it with TLR ligands in culture, and then stain the cells to do flow cytometry. So then we can analyze the expression of different cell types within the circulation, as well as activation markers, including class 2 on those different cell types. So, Angelique.
This may be slightly off topic, but when you're collecting blood samples, I noticed that the tubes usually have some type of, I don't know, other something else in it before you put the blood in. I don't know what is it and what is its significance.
Anybody have an answer?
Is it anticoagulant?
Anticoagulant, yeah.
Okay.
So it depends on if the tube is designed for serum or cells.
Mm-hmm.
And if it's for serum, then it promotes clotting. And if it's for cells, then it has different anticoagulants in it. So, and then the different downstream processes are tolerant of different anticoagulants. There's heparin, BTA, etc. So this is an assay I was performing a few years ago, and we had a hypothesis about the involvement of a TLR ligand in activation of cells from lupus patients. And so what we wanted to do was use drugs to try to block TLR activation. And so this was setting up a whole panel of markers to identify the different cell types in circulation. So using light scatter, it's pretty easy to identify lymphocytes, which are small and don't scatter a lot of light, which means they don't have granules, versus neutrophils, which scatter a lot of light because they have granules. So the first basis on which the cells get separated is on their light scatter characteristics, and then we identify B cells as CD19. So you can see in whole blood that can be about 1 to 5% of the circulating lymphocytes. Likewise, we use CD3, we mentioned earlier, to identify the T cells, and then CD14 to identify the monocytes. And here I'm looking at the expression of HLA-DR, so one of those class 2 molecules I was just talking about. And HLA-DR is very high on antigen-presenting cells, dendritic cells, and monocytes. And then if we go down here and gate out the B cells and T cells, we can identify another cell type called NK cells in circulation. So this is basically a way to simultaneously, in a single blood sample, identify all the major white blood cell types and look at class 2 expression on the surface. And so we stimulated the whole blood with a number of different TLR ligands, CpGs, which are TLR9 ligands, and RH4H, which is a TLR7 ligand. You can see in the professional antigen-presenting cells, monocytes and B cells, the exposure to those TLR ligands increased the expression of class 2. So they already had a lot to begin with, and they express even more after stimulation with the TLR. NK cells have no receptors for those TLR ligands, so they have almost no response. So this is just basically recapitulating in a test tube what happens when you have an infection. The exposure to components of that bacteria or virus are sensed by receptors of the innate immune system, and you can increase the HLA-DR so that you're more likely to be able to present foreign antigen. So then we went on to study our drug, which we were hoping was modifying this upregulation of class 2. Okay. So polymorphism within an individual allows presentation of most peptides. Diversity of the genes across the species allows natural selection in response to new pathogens. So again, going back to what Jenny asked about, each pathogen is going to try to make some molecule that can't be presented to the immune system, so it's invisible to the immune system. So there may be holes in an individual's ability to present a given peptide, so no MHC that can bind that peptide, or no TCR specific for MHC peptide combination. So there's been continuous evolutionary pressure to increase TCR diversity and to increase MHC copies to be able to present every potential peptide. So certain MHC haplotypes are predisposed to certain autoimmune diseases. So if you have certain MHC haplotypes, you might be more likely to present some self-peptides in a way that activates the immune system. So these are complicated inheritance patterns. People don't inherit these in a dominant Mendelian pattern, but they inherit a pattern of HLA that increases their risk of autoimmunity, but it doesn't cause autoimmunity. So HLA haplotype can provide a genetic or immunological background which increases the chance of autoimmune disease in a given environment. So again, the chance that somebody develops autoimmunity depends on their genetic background as well as the environment they're exposed to, including infectious diseases. So this just shows you that depending on the HLA allele, you can be predisposed to a whole bunch of different autoimmune diseases, including diabetes, arthritis, Graves' disease, multiple sclerosis. So these are all tissue-specific autoimmune diseases. So you can imagine that these specific HLA alleles present some peptide from those tissues in a way that activates the immune response. Okay, we did it.
Didn't make it,
She's
so
worn out. Yep. I don't know who's more tired, Colin or Jenny. Okay,
That's a good question.
so the TCR is similar structurally and genetically to the PCR. TCR diversity is driven by genetic recombination just like that of the PCR. The TCR can only interact with peptide antigens and only those in the context of MHC. I'm not going to say the next two because I've said it enough times already. Class 1 and Class 2 are expressed at different levels on different cells. Unlike the TCR and the BCR, we have many different MHC molecules on each cell. MHC expression is genetically diverse within and between individuals, driven by infectious disease pressure. However, the byproduct of that evolutionary pressure is MHC genes that can increase the chance to react to a self-peptide. And a lot of the, next week we'll do T-cell development and T-cell mediated immunity, just like we did with the B-cell. And then we'll be done with the mechanics, then move on to diseases. Good luck with your exams. Don't overdo it. I'll see you next week. I probably